Are you being able to see my slides?
Yeah.
Okay, then let's get started. Good evening, good afternoon, and good morning, everyone. We have people joining in from all over the world, and that's why greetings to all of you. I am Sailing Ray. I'm a member of Jnana Prabodhini Foundation's iBRETA team, and it is my great pleasure today to welcome you all here for the third session of Health Katta series. If you have missed our first two sessions, please find our videos on YouTube on Jnana Prabodhini Foundation's YouTube channel. I'm sure you'll find them insightful. With that, let me start by introducing Jnana Prabodhini Foundation, or JPF for short. In and around 2019, a handful of Jnana Prabodhini alumni and associates who have been settled or have been studying abroad came together with a dream of giving back to their institution and to their country. With multiple brainstorming sessions, JPF was born. JPF is a 501c3 non-profit organization registered in the United States, which has the same values aligned with its parent organization, Jnana Prabodhini, or JP. JPF focuses work in mainly three areas: rural development, education, and women leadership. JPF in its past 3 years has conducted numerous different activities like drought relief, energy sufficiency, women entrepreneurship and empowerment, and talent development, sponsor a child program, and teachers trainings programs. Currently, we have over 750 individuals from 12 different regional chapters spread across the US, Europe, and India. And we have more than 200 and more than 100 active volunteers joined us for this mission and vision. Today, I will be introducing the iBRETA team. So among the plethora of projects that JPF has, iBRETA is one of them. iBRETA is short for Initiative for Biosciences, Research, Education, Training, and Awareness. As the name suggests, iBRETA is a platform that covers 4 different aspects: research, education, training, and awareness. Under the research aspect, we aim to conduct projects that will develop an investigative mindset among researchers in the field. With education aspect, we want to induce scientific acumen of students by providing them with a skill-based approach for tackling a scientific question. Under training, we want to become a tool for people to seek expert guidance and training for managing important issues in public health. And under awareness, we want to become a bridge for enhancing communication between the medical community, its field experts, and the general public to provide authentic scientific information about health and diseases. Today, iBRETA is proud to have an eminent speaker, Dr. Yogesh Shouche, for our third Health Katta session. And it is my honor to introduce him to all of you. Dr. Yogesh Shouche was a research scientist at National Center for Cell Science, NCCS Pune, which is an autonomous institute under the Department of Biotechnology, DBT, Government of India, for more than 30 years. He is currently a professor at Azim Premji University at Bangalore. His area of interest is microbial community in unique ecosystems and how they interact with each other and with the surroundings. At NCCS, he developed microbial culture collection, which is the largest in the world under one roof, and is also International Depository Authority, IDA, under Budapest Treaty, and designated repository under Biological Diversity Act 2002 of India. He has worked on multiple interesting ecosystems such as effluent treatment plants, impact of Kumbh Mela, which is the mass bathing event on river ecosystems, and stratosphere. So from the earth to the clouds everywhere. In the past few years, his group has worked extensively on human microbiome project of Indian population and how it is affected by the lifestyle and genetics. Apart from that, he has also conducted some intriguing studies to understand microbial resistance to antimicrobials and antibiotics. He has keen interest in science popularization and has written many articles that can pique anyone's curiosity in microbiology. He's also an avid speaker. He has delivered multiple speeches in schools, colleges, and universities, and other public events. Today, we are honored to have Dr. Shouche with us to explain how gut microbiome affects our lifestyle and how our lifestyle in turn affects our gut microbiome. So, Dr. Shouche, please go ahead and enlighten us.
Yes, so thank you very much for this nice opportunity, and it's
I think, sir, you are mute.
yeah. I wanted to put the video, but by mistake, yeah. So now this is fine. Yeah. So I'll share my screen. So it's really my pleasure and honor to be talking to all of you today about microbiome. So first of all, I mean, I would just like to introduce you to the concept of microbiome. So what is made by a microbiome? We often hear this word microbiome, but before we get into our talk, we should first understand what is a microbiome. So the word microbiome comes from microorganisms, and these are living organisms, microscopic organisms which we cannot see with our own eyes, and they are present everywhere. They inhabit all environments, and the environments which we otherwise cannot think of having a life, like rocks, marine sediments, so on and so forth. So they inhabit all these places, and they are under different categories: bacteria, fungi, viruses, and protozoa. So these are the major categories of microorganisms. But normally when we talk about microorganisms, it's mostly we are talking about bacteria, but it includes other forms as well. So microbiota is the community of organisms living in a specific environment, and microbiome is a community as a whole. And the practical difference between microbiota and microbiome, as I'll explain you later, is microbiota is something which you can grow in the laboratory, and microbiome is the total component which cannot be grown and can be grown in the laboratory. So this part will become little more clear in a later part of my talk. But point to remember is microorganisms are present anywhere, everywhere, including living organisms. All living organisms contain microbes associated with them, which constitutes a microbiome. So whether it is plant, animal, even eukaryotes like protozoa, they all carry a specific group of microbes with them, and that becomes their microbiome. Why specifically human microbiome has assumed importance in last couple of years is because it has extraordinary human health implications, which again we will see towards the end of our talk, that for that reason it has become very important. And it is believed, or it is not just believed, it is already there as a fact, that it provides simple, sustainable and safer solutions for human life, not possible otherwise. And it offers a wonderful tool for personalized therapies. So that also we will see some examples of that. And many Nobel laureates have emphasized the importance of microbiome. So this is Joshua Lederberg and then Julian Davies. They all treated this as a biological revolution of 21st century. And they said that the microbiome is a very important component of human body. So when we look at our body as such, the thing that comes very strikingly is if we count the number of cells in our body, the microbes outnumber in terms of number of cells, they outnumber human cells in our body by several folds. So earlier it was estimated that the number is 10 times more. The current estimate says that 2 to 5 times more, but undoubtedly the number of microbes present in our body, different parts of the body, is much more than our own human cells. So that would emphasize the importance of and role of microbes in our body. And if you talk in terms of genetic content, then whereas human genome today is known to contribute something like 23,000 functional genes, the whole microbiome put together contributes several thousand genes to our body. And that will be evident by the kind of role microbiome has in our health. So this number of genes has tremendous impact on our health. So the major organs that carry microbes are obviously the gut is the most important microbe, which carries a large density of microbes. The total weight of microbes present in the gut is something like 1 kilogram. Then you have nose, then mouth, vagina, skin. So all these, and this is just a partial list. Many other organs carry, for example, lungs, then ears, upper respiratory tract, vagina is already mentioned, their placenta. They all carry their own unique communities of microbes. And important thing to remember, especially about gut and skin, is that when we say there are microbes present on these organisms, obviously it's not a uniform distribution. So when we talk about skin, the different parts of the skin, like our forehead, which is oily and exposed to outside air, will carry different microbes than armpit and groin, which are moist and not exposed to outside air. And gut is like I would say like a globe or a nation, because it's the starting from the stomach to large intestine and the anus, the type and density of microbes varies tremendously locally. So it is not that when we say that X number of type of microbes are present in the gut, it doesn't mean that they are uniform. Stomach, obviously being acidic in nature, carries acid resistant organism, but even large and small intestine, the microbial distribution is not uniform. And there are different pockets where the microbial densities and microbial nature of the microbes varies. So this is a point which we should remember. And obviously we know very little. The variation of microbes within the local variation of microbes within the gut, we hardly know anything about that. So how do you study the microbiome? And the answer to how you study the microbiome lies in this observation in microbiology, which was done several years back. What the observation is a very interesting observation, that not just human gut, but you take any sample, you take soil or water or your tooth scraping, any sample you take, put it on a slide and observe it under the microscope and count the cells per ml. You will see, suppose you see some 10,000 cells, the same sample you try to grow it in the laboratory using the kind of culture media that are used to grow microbes in the laboratory, you will get only 100 cells growing here. So what happens to 9,900 cells? those 9,900 cells we are unable to grow in laboratory, come what you do. The reasons are many reasons. The primarily we are unable to reproduce the conditions that support their growth, and maybe they cannot grow in isolations, they only in groups. So the reasons are many and we don't know much about that. But the fact remains that if you get 10,000 here you get only 100. So there was no way to study these 9,900 for several years. But what happened was there were many developments which helped, and this was one development that happened sometime in 1970s, that this particular person, Carl Woese, proposed that ribosomal RNA gene serves as a molecular clock, and looking at its sequence, you can classify and identify the organism. So this was a very important development, because here it's not the growth of the microbe, but a molecule present inside the cell helps you in the identification. So that was the milestone number 1. The second milestone was the revolutionary discovery of polymerase chain reaction, wherein you can amplify a single copy of DNA several million times. So this serves many purposes as far as microbial studies are concerned, because then you may not be able to grow the microbe in the laboratory, but you can amplify the ribosomal RNA gene and study the gene. That is point number 1. The point number 2 is, as the PCR technique goes, a single molecule over the 32 cycles is amplified exponentially, and if there is a single molecule at the end of 30 cycles, you get million copies of that. So which also means that in any environment, whether it is gut or soil or whatever, if there is a single microbe per ml or per gram, theoretically when you do PCR, you can get million copies of 16S ribosomal RNA gene from that organism. So even the organisms that are present in the smallest number can be detected using PCR. Now, obviously, when you do a PCR from a mixed population where there are 10,000 microbial species present in the soil, your PCR molecule will contain mixture of 10,000 different sequences. Now, how you separate those sequences is a different story. I will not go into the details of that. Earlier, different technologies were being used for that, but now we have these NGS technologies, which is called as next generation technologies. They use a very specific way to segregate these 10,000 different sequences types from each other, and then sequence each of these 10,000 different get information out of this each of 10,000 unique sequences. Each of this technology uses a different technique to do that. I'm not going to go into the details of that. Somebody who is interested can either find out about this technology or ask me a question, and I'll try to explain it. But point to remember here is that these techniques, listen to my word carefully, that these techniques quantitatively amplify the sequences from the sample, and this quantitative amplification and sequencing is very important, because at the end of the study, when you get the sequences of the microbial community present in any sample, it also tells you the relative proportion. So when there are 10,000 species, when I say there are 10,000 species of bacteria present in the gut, and we get the sequence in any of these 10,000 species, sequence I get in any of these sequence technologies, it is quantitative in the sense that the species which are present in larger number, you get larger number of sequences from that. Species which are present in smaller number, you get less number of sequences from that, and that is proportional. So if some organism is present 10 times more, you will get 10 times more sequences. So important to remember is that technique is quantitative, and this quantitative nature of the technique has helped tremendously in doing comparative microbiome studies. For example, the comparison of quantitative comparison of microbiome in healthy and disease status. So immediately, since the technique is quantitative, you can find out when you get a certain disease, which type of bacteria are increased in number, to what extent, which type of bacteria are reduced in number, and to what extent. So that's the importance of these techniques being quantitative in nature. And probably at the beginning of this century, this would not have been possible because the sequencing costs were very high. A single human genome sequence was supposed to cost 15,000 crore rupees and take 11 years of time. But the technologies mentioned here, these are all faster. They are called as high throughput sequencing technologies. So they are faster and cheaper and generate huge amount of data. So although prima facie, the technique may appear to be very expensive because some of these technologies, the instruments are costly. They cost, in Indian currency, they cost few crores. And in Indian currency, a single run on this equipment cost at times few lakh rupees. But eventually the amount of data which you get makes the technique cheaper because per sample cost is very less. So that's how, since the per sample cost reduced dramatically to several fold, bring it down to $100 per human genome sequence and time less than 24 hours. Even microbiome studies were possible because if you compare the size, the human genome is equivalent to 50 to 60,000 bacterial genomes. So the time and the cost in which you can do a single human genome, you can do 50 to 60,000 microbial genomes. And that's the reason all these microbiome studies were possible. And in the last 2 decades, huge amount of information has been collected using these high throughput technique. It is called as metagenomics, and it is applied not only to human, but it has applied to all the ecosystems, including soil, animal, and extreme environments like glaciers, Antarctica. Literally thousands of studies are available where these techniques are applied to do the comparative microbiome studies. There are many ways in which you can look at these microbes. One way, as I said in the beginning, is you can just sequence a typical bacterial cell has something like 3,000 genes. So out of these 3,000 genes, you sequence only single gene, that is 16S rRNA gene, which is important for taxonomic purposes. So since it is important for taxonomic purposes, it gives you taxonomic picture. So it gives you taxonomically what are the different type of organisms that are present in your sample. Many times this information is useful and it gives you valuable data, but with the cost of sequencing going down, it is now also possible not to depend on just one gene, but sequence all the 3,000 genes for an organism. So which means that you do sequencing to such an extent for your sample that if you have 10,000 different species, probably all the 10,000 species, you have the whole genome sequence. So you do the sequence to that extent, you generate huge amount of sequence data, and then you mine that data using bioinformatic tools to understand the functional aspects. So bioinformatically, you can assign the functions to the sequences and find out what does the microbial community in a given sample exist in terms of functional attributes. From the genome sequences, you can predict the functions. You can only predict. You actually don't know what is happening, but you can only make predictions. In order to confirm what is actually happening, you use these other techniques which are called as transcriptomics, where you sequence RNA that is expressed under different conditions. You look at the proteins using proteomics, or you study the complete metabolites, metabolite composition under different conditions, and this is called as metabolomics. And today omics has become a very popular word. There is a lipidomics and so many omics. So but these are the major techniques that are used to study the microbiome structure as well as function. So when we talk about human microbiome, it's not constant. As we are born, it is believed that the baby in uterus is sterile, and the first exposure comes at the time of the birth through vagina, and then the breast milk gives the not only microbial inoculum but complex oligosaccharides present in the breast milk. They function as a prebiotics that promote the growth of only specific good bacteria in baby's gut. And once the baby stops having mother's milk, then the food and the other environment affects the baby's gut microbiome. So the microbiome develops from the birth to infancy and childhood. These are the factors that affect the microbiome. And as I said, that as you grow, the microbial diversity increases. At the adulthood, there is a maximum diversity. And then as you start growing older, again, the diversity starts decreasing. The microbiome picture is different in the elderly persons above 60. And today, a lot of research, microbiome research, is going into the role of microbiome in healthy aging and whether we can promote healthy aging by altering microbiome. So that's a very important topic of research right now. And this is something which I already said, that the baby gets microbial inoculum from vagina, then through the breast milk. And if the baby is born by cesarean section, then it does not get microbe from the mother, it gets microbe from the environment. Matters become worse if the baby is not breastfed, and such babies are subsequently prone to have diseases like allergies or diabetes and obesity. So I have already made this passing remark on this point, that the microbial density varies in GI tract, stomach having the lowest and colon having the largest. But even within, when I say colon is 10 raised to 10 to 10 raised to 12 CFU per ml, it's not throughout uniform. Community as well as density is not uniform throughout. There is a tremendous local changes. So these are the things which I have already mentioned, how it is colonized at the birth, it develops in early life, and it is very, very individual specific. So much so that there have been people in the literature, people have shown many forensic applications of microbes. It is generally stable throughout the life and affected by transiently affected by internal and external factors, and alterations in the disease is something which we are going to see in little more detail. So it has variety of functions. It helps in the development of organs, epithelial turnover, immune system maturation. Most important function is energy utilization and storage, synthesis of micronutrients and short-chain fatty acid. This is a very important functions, block colonization with pathogenic bacteria. And today, lot of molecular details are available as to how the colonization with pathogenic bacteria is blocked. The orally taken drugs are also metabolized by microbes, and lot of drugs are made depending on microbiome. Either they are made inactive in the gut or their activity is increased. A lot of research is going on that. Today, there is a very strong evidence for gut-brain axis through microbiome, and microbes in the gut are also known to affect brain function, and thus they are correlated with many neurological disorders, memory, mood swings, depression, anxiety, intelligence. All these are associated with gut microbiome today, and there are hundreds of studies on this. So it is very difficult to what is defined as a healthy human microbiome. It is different for definitely you cannot define healthy human microbiome universally. So the definition of composition of healthy human microbiome is not universal. It varies depending on your ethnicity, your diet, your biogeographical location, and many other factors. So each individual probably has his standard healthy microbiome, which gets altered during the disease. So I mean, and all the organs have gut, skin, vagina, mouth. All these organs have its own healthy, what you can define as healthy microbiome, and it gets altered during the disease conditions or dysbiosis. So there is no universal healthy human microbiome composition. It is specific to a particular group, or even I would say it is specific to a particular individual. So globally, there is tremendous variation in the microbiome. The studies have shown tremendous variation in the microbiome, and each country has its own unique standard microbiome. India is again a very different case because we take a pride in saying that we are as diverse as the entire globe. So within India, there is tremendous variation in microbiome, of which we have not understood much. So today, when we talk about factors that govern human health, earlier we used to talk about host genetics and lifestyle. Today, we also include microbiome as a component that affects the human health. And diet is one of the important factor that affects the microbiome. So diet has, I mean, the interaction between diet and microbiome is very complex because and health is very complex because on one hand, the diet directly affects our health, and it also affects our health by altering the microbiome. So when we say a good diet is not on, it's good for us in two ways. One way, it's really good for our health. It has our health promoting benefits, and it also promotes the growth of good bacteria in our gut. So the good diet has multiple effects, and also the diet affects the gene expression in our intestinal cells, and that way again it affects our health. So the interaction between host, diet, and microbes is a very complex interaction, and again we have started to understand little more about this interaction in last couple of years. So though the what constitutes a healthy microbiome, the definition is I mean the composition is different for different individuals, but there are, in general, there are some bacteria which are thought to be health promoting and known to be affected by the diet, and this is a list of some of the bacteria which are commonly affected by the diet and their physiological role in our gut. So there are bifidobacteria, lactobacilli, bacteroides, Alisepsis, Bilophila, Clostridium, Roseburia, Eubacterium, Enterococcus, Faecalibacterium, Akkermansia, E. coli, Helicobacter, Streptococcus. So these are all somewhere or the other in the literature, they have been shown to be affected by diet. Many foods also affect the diet, and I'll talk about this in little more details in the So there have been studies which have shown the effect of diet on the gut microbial composition, and so there was one study where it was shown that if you consume plant proteins, there is an increased abundance of bifidobacteria, lactobacilli, decreased abundance of bacteroides and Clostridium, and increased production of short chain fatty acids, and increased gut barrier and tracts, and reduced inflammation. As against that, if you consume animal protein, the bifidobacteria decreased, Ruminococci, Bilophila, Aliseptis, and Bacteroides increase, and there is a decreased production of short chain fatty acids, increased production of TMAO, which leads to cardiovascular disease, and reduced short chain fatty acid leads to development of inflammatory bowel disease. Within the, among the fat, unsaturated fat and saturated fat. So unsaturated fat increases Streptococci, Lactobacilli, Bifidobacteria, Akkermansia, and saturated fat increases Bacteroides, Bilophila, Faecalibacterium, Prosnitza. So the fat composition also has effect on diet, and then these bacteria in turn are responsible for TLR inactivation and inflammation, and in turn they have effect on cholesterol in our body. Increased bacteroids and Bilophila also reduced to decreased insulin sensitivity. So diet, gut bacteria and physiology, there is a, I'll skip some of these slides. There is a very complicated interaction between us, and the Western diet, there was a comparison of Western diet and Mediterranean diet, and Western diet was of course found to reduce the abundance of Bifidobacteria, Lactobacilli, eubacteria, and increase bacteroids and enterobacteria. So which in turn leads to inflammation and increased risk of diseases like cardiovascular disease, inflammation, and diabetes, and obesity. So Western diet here means the diet that is high in fat, low in fiber, and high in sugar. And Mediterranean diet is supposed to have health, is shown to have health beneficial effects. Again, I'll skip this slide. So when we talk about microbiome and disease, it is believed that there are 3 types of bacteria in the gut: symbionts, commensals, and pathobionts. And as long as they are in the harmony and there is a homeostasis, we are healthy. If this balance is disturbed by variety of factors, then pathobionts increase and dysbiosis results. And the factors that affect is of course some genetic factors, then microbiota modulators like antibiotics and pathogens, and environmental factors. As you know, as we discussed, that the diet is being the most important factor which increase the proportion of pathobionts and the dysbiosis and the disease results. And many diseases, now the Microbiome Association has been shown, but this list is incomplete. Now today we know many neurological disorders like Parkinson, Alzheimer, also there is a microbiome role. So these are the physiological functions of the microbiome, and these are the features. Like when you have disease, there is a insulin resistance, gut inflammation, LPS production, increase TMAO production, increased TMAO production. All these are indications of health, and these are typically done by influenced by higher sugar intake, excessive protein consumption, saturated fatty acid intake, and proton pump inhibitors and antibiotics. They all promote bad bacteria and have all these effects. As against that, dietary fiber and probiotics, I would recommend natural probiotics, natural fermented food. They all promote the growth of good bacteria and have all these health beneficial effects. So the probiotics and dietary fiber are the essential components that help the maintaining healthy bacteria and good gut health. And when we look at the microbial association with certain physiological disorders, I'm going to take example of obesity and diabetes. So this was a classical study done almost 10 years back, where the microbes were taken from an obese person, put in the mice, and mice become obese. Microbes were taken from lean individual, put in the mice remained lean. So this was a classical study done almost 10 years back that showed association of microbes and obesity. After that, many experiments were done to show microbiome and obesity link. And today what we understand is that, again, as I said, that the complex relation between diet and microbiome. So in addition to genetics, the consumption of energy dense food, sedentary lifestyle, hygiene, and medication, all these affect microbiome, and the result is decrease in microbial diversity, decrease in microbial gene count, decrease in organisms like bifidobacteria, Oscillospira, Akkermansia, increase in Roseburia, Prevotella, and this results in adiposity and dyslipidemia. However, I want to give a word of caution here. These changes are population specific. So typically when we talk about Prevotella, the Western and Asian population, Prevotella is shown to be associated with obesity and diabetes. As against that, in Indian population, Prevotella is shown to be associated with health, and decreased abundance of Prevotella is associated with diabetes and obesity. So these microbiome changes are also region specific or community specific. So these are the microbial taxa that are altered in obesity. The Firmicutes bacteroides ratio is altered. Firmicutes abundance is higher. Proteobacteria, molecules, and lactobacilli, there is increased abundance of this, and decreased abundance of Akkermansia, Faecalibacterium, Bacteroides, Methanobacterus bithi, and Lactobacterium plantarum and Paracase. So decreased abundance of these. And this results, I have already shown that higher abundance of bacteria that ferment carbohydrates, leading to increased rates of short-chain fatty acid biosynthesis and extra source of energy for the host, which is eventually stored as lipids or glucose. Increased intestinal permeability to bacterial LPS, resulting in elevated systemic LPS that aggravate low-grade inflammation and insulin resistance. And then finally, increased activity of gut endocannabinoid system. So these are the effects of altered microbiome in obesity. Same way for diabetes also, microbiome association has been shown. Again, this was a classical study again done some 10 years back, where the women have a condition which is called as gestational diabetes. So first trimester, same woman healthy, microbiome put in the mice are normal. The same woman becomes diabetic in the third trimester, microbiome put in the mouse become obese and show insulin resistance. So this was a classical study done way back in almost 10 years back. And today, again, a lot of studies have been done, hundreds of studies, and overall picture that emerges in gut microbiota diabetes association it has been proven in animal models also. And microbiome changes are not just associated with diabetes, but the studies have been done on diabetes associated retinopathy, cognitive impairment, macrovascular complications, diabetic nephropathy, diabetic induced reproductive system damage, and peripheral neuropathy. So microbiome changes associated with these complications also have been studied in last decade in relation to diabetes, and a wealth of information is available. So again, a general picture that emerges from the available studies is decreased in Clostridium species, Akkermansia, Faecalibacterium prosbisci, Clostridium fusobacterium, and increased abundance of Velionella, Lactobacilli, Bacteroides vulgatus, E. coli, Clostridium, and different species of Clostridium. But again, I would like to warn that these are these observations may not be applicable to India, because in India we see that the microbiome picture, the healthy microbiome picture itself is different. So these results are again population specific. The data presented here is mostly the studies from Western population, USA and Europe. So these are generalizations for these population, and these are some of the major findings. I'm not going to go through the individual findings, but I have just summarized some of the individual findings here, that the kind of strategy that was used, either 16S rRNA gene sequencing or metagenomics, the population that was studied, and the main observations coming out of that studies. I am not going to read out all this. the point to emphasize here is that a lot of studies on different populations have been done for diabetes also. And so there is a alteration in not only during the diabetes gut microbiome is altered, but also skin microbiome also is altered, and the signal from altered gut microbiome goes to brain, and from brain it goes to skin, and the compounds that are secreted in the skin are altered, which in turn again affects the gut microbiome. So it is not only the gut microbiome that is affected in diabetes, but even the skin microbiome is also affected in diabetes. So how do you maintain the healthy gut microbiome? Obviously, it's abundantly clear from all that what I've said is food with high fiber, low fat, and low sugar promotes the healthy microbiome. Probiotic preparations are useful to a certain extent, but the best way is to consume traditionally fermented foods. Avoid processed foods, even, I mean, any kind of processed food with preservatives is detrimental to healthy gut microbiome. And there have been studies that have shown that the consumption of junk food results in irreversible alteration in gut microbiome. So up to a certain extent, if you shift from junk food to healthy food, the microbiome reverts back. But if you continue to abuse yourself with junk food, then the microbiome, it's a no point of no return, and the healthy microbiome never comes back. This has been shown in on the studies on tribal gut microbiome. So the studies have been done on the tribals who migrated, I mean, from their traditional localities to bigger cities. So cross-sectional studies were done and shown that those who are residing in cities for long period of time have lost their healthy microbiome. And in experimental mice model also, it was shown that after a certain number of generations, if the mice are consistently given low fiber, high sugar, high fat diet, then their microbiome, healthy microbiome is completely lost. Antibiotics are known to alter the microbiome. So take antibiotics only when necessary, and only and complete the dose. So don't because this leads to other complications of developing antibiotic resistance. So complete the dose, and don't stop it halfway through. The final point is it's a question whether to what extent you should follow the hygiene and cleanliness, because it has been shown that the more you get yourself exposed to other microbiomes, you enrich your microbiome, it becomes more diverse, and more diverse microbiome is healthy. So unfortunately, during the corona, we had gone to the extreme of hygiene, but to what extent you should go for hygiene, having everything disinfected and floors cleaned with disinfectant and disinfected soap, to what extent we should go is a big question mark. To a certain extent, not following too much of hygiene increases your diversity of your microbiome and in turn your health. So there are many major microbiome initiatives across the globe. Most of these the studies have yielded tremendous information about microbiome and disease associated variation in microbiome for those countries, and this information is being now used for precision medicine. So for the this diet microbiome association is being used for the prevention of the disease, diagnosis of the diseases, therapeutics, and personalized nutrition. So it's a very important and upcoming field to use microbiome information and microbiome diet association for the precision medicine in the areas of prevention, diagnostics, therapeutics, and personalized nutrition. The best way to do microbiome based is the crudest way is the fecal microbiota transplantation. but that is again an old story now. We have moved from crude fecal microbiota transplantation to microbial consortia to single isolates to genetically engineered microbes. So far have not been used that extensively, to bioactive molecules. So from the healthy gut microbes, people have isolated bioactive molecules which are being used in therapy today. So it says the same thing, that as you go from crude fecal microbiota transplantation to bioactive compound, the specificity of your treatment increases, and the ecosystem effect also decrease as you move towards the specific compounds. So this is just an example of the currently available molecules isolated from members of microbiome. These are under clinical trials. So you can see phase 1, phase 2, phase 2, phase 1, phase 1, phase 3 clinical trials. So these are the code names of the compound and the company that has produced them and the diseases for which these are being used. So recurrent Clostridium difficile infection, ulcerative colitis, metastatic melanoma, obesity, cancers, Clostridium difficile. So different purified compounds, bioactive molecules derived from microbiome are currently under different stages of clinical science. So the point to emphasize is that the microbiome science has moved far away in last 2 decades, and we have reached a stage where we have actually biomolecules, bioactive molecules available from the members of the gut microbiota, and that makes the global microbiome market as a huge market. So it is projected to be $1,370 million in 2029, and in variety of areas, in the therapeutics as well as diagnostics, the market share is almost the same, and these are some of the key players in microbiome based therapeutics. So I think, and these are the 5 major microbiome companies globally: Vast Biome, Boost Biomes, Folium Science, Anisome, and Reba Analytics. What I also want to emphasize here is that these 5 top companies, not all of them are working in human microbiome. Some of these companies are working in animal microbiome and agricultural microbiome. So not just human microbiome, but animal and agricultural microbiome is also a very, very challenging and upcoming field. With that, I'll stop here. Thank you very much. Thanks for your attention. And I think we have ample time to take any questions. Thank you very much.
Thank you, sir, for such an insightful session. And it was intriguing to know that how so many tiny creatures which are living in our body are either our friends or our enemies, and how our simple lifestyle changes or like dietary changes could help us improve the good bacteria that we have. So we have received like a lot of good questions even before the session was there. I'll try to cover as many as I can.
Sure, yeah.
But
and those that cannot be answered here, I wouldn't mind answering them over the email. I mean,
yes,
either
sure.
you can forward or the persons can drop me a mail and I'll reply to the mail.
Sure. Yeah, and we'll also like compile all the questions that we have got,
Sure.
and then you could
Yeah.
answer, and we'll email
Yeah.
everybody. So
Right. Yeah.
I'll still try to do justice to all of the questions. So you talked about how different environments have different gut microbiome development. So one of the questions was how does it differ between rural and urban areas in India? Like for now, India, because it changes with different territories as well. So is there any study which tells like are there any similarities or differences between rural gut microbiome and urban gut microbiome?
Specifically rural and urban gut microbiome differences, there are no studies. See, if you look at the India, the number of microbiome studies are very small. I mean, when in other Western countries, there are thousands of studies. The India, you will not find the number will not even go to 3 digits, if the total number of studies that are seen. So this,
Right.
I mean, we ourselves had done a study where we compared the microbiome of semi-urban and urban areas, and we definitely found the differences in the microbiome. But that was only one study. The larger study has not been done to draw any conclusive conclusions. However, currently a bigger microbiome studies is underway, which involves doing some microbiome for some around 3,000 individuals. And with that number, which is done across 4 biogeographic regions in the country, that would give us some more conclusive information about how the microbiome changes in rural and urban settings.
Okay. Thank you. I hope that study gets done quickly so we know
Yes.
more about our gut microbiomes. So you also talked about how probiotics are good for developing good, healthy microbiome. And there was a question with like, how do you know what kind of microbiomes are taken in when you have prebiotics and probiotics, and what's the difference between those two?
No, can you? I didn't get the question. What is the question?
So what's the difference between like prebiotics and probiotics, and what kind of probiotics is good? Like, how do you decide what individual person should take what kind of probiotics?
Okay, so the difference between probiotic and prebiotic is very simple. Probiotic is a live organism which you take in the form of capsule, tablet, or in the form of preparation. Today, you also get probiotic ice creams and such drinks also, so you can have probiotics through them. And prebiotic is the compound which promotes the growth of healthy bacteria in your gut. So that is the difference between prebiotics and probiotics. And my take on probiotics is there is no organisms which can be called as a universal probiotic. In my opinion, considering the variation in gut microbial population across the globe, good probiotic should be population specific. Otherwise, it will not have its effect.
Right.
So,
So is there any study? I know there are less studies in India, but do we know like in Indian population what kind of bacteria are good, or what is a better probiotics that are there?
see, all the studies done in India, whatever smaller number of studies are available from India, it clearly indicates that against, I mean, in contrast to what is observed in Western population, the Prevotella seems to be the indicative of health in Indian population. So the healthy individuals have higher abundance of Prevotella. So that's the overall picture that emerges. And if you have diabetes or if you are obese, the Prevotella abundance decreases.
Okay.
So in my opinion, the probiotic for Indian population should come from Prevotella and not from lactobacillus or something. So people are trying to generate isolates of Prevotella and then study their health beneficial effects so that indigenously a Prevotella probiotic can be developed. It's an anaerobe, so it's very difficult to grow actually.
Oh, okay. Thank you. And you also talked about various diseases that we observe, which are in relation to different gut microbiomes. And we know that you have a paper published in which you have studied how cancer and microbiomes are related. Could you tell a bit more about it?
See, cancer again in India there are no studies on cancer and microbiome, but if you look at globally, then there are many studies, especially the cancers like I'm not getting the name, but oh, it's the cancer of anus. I mean, I'm not getting the colon cancer. Sorry. So the colon cancer microbiome. There are a lot of studies on colon cancer and microbiome, but those studies cannot be extended to India because the nature of colon cancer in Indian population itself is different. So, but in India, there are no studies so far on association between cancer and the microbiome.
Okay. And with regard to the previous question, do we know are there any food products that consumption of that would increase the Prevotella microbiome?
See, again, there are no studies on that, but what
Okay.
has been shown in earlier is that the Prevotella is associated with high fiber intake. So obviously
Okay.
the high fiber intake, and in our studies also we have seen that the high fiber intake is associated with higher abundance of Prevotella. So intake of high fiber food would promote the growth of Prevotella.
Right. And you also talked about how diabetes and microbe relation has been studied. So one of the questions was like, we know that in India there is a good high percent of diabetes in population. So how much is it related to gut microbiome that we have, and how much is the genetic factor? And is there any way that we can have a particular kind of diet or lifestyle which would help us reduce that?
See, diseases like diabetes are multifactorial diseases. So they have
Right.
genetic component and they have a gut microbiome component also. Now, how they interact with each other is very difficult to decipher. But my take on this is what is being done currently is through longitudinal studies or even cross-sectional studies. Even we have also done that you look at the microbial composition in the healthy individuals, pre-diabetic, diabetic. So that would give you some idea as to how the microbiome is getting altered as you move towards diabetes. And the idea there is if you are able to capture the individual at the pre-diabetic condition, the microbiome alteration, then you can use probiotic, prebiotic or some kind of microbial therapy to delay the onset of the disease by giving probiotics. Or even the person with diabetes, if you use microbial therapy, diabetes cannot be reversed. But in my opinion, the dependence on the drug can be brought down. So either the drug dosage would increase, or if somebody is taking injection, the dose of injection would increase, or instead of injection, the drug may suffice. So that kind of intervention is possible, but I wouldn't say that we would be able to stop the medication altogether using microbes. It will reduce the dependence on the drugs and probably delay the onset of the disease also if we start the microbiome therapy right in time.
Right. Yeah. Thank you. And you also talked about how immunity is also either developed or reduced using gut microbiome. So what all aspects of immunity and how autoimmunity also has a role with respect to gut microbiome?
See, role of gut microbiome is in autoimmunity. I mean, there have been some hypotheses about diabetes itself, that the gut microbiome in some way is responsible for autoimmune diabetes, but I don't remember the exact details of that. But otherwise, the the accepted belief is that the gut microbiome kind of trains the immune system, and one hand it trains the immune system, on the second hand, as I mentioned, that it inhibits the colonization by pathogenic bacteria. Gut bacteria produce certain compounds that prohibit the pathogens from colonizing the gut, and also there is a physical competition. So if the same location in the gut, the gut bacteria do not allow the pathogen to come in, and when you give antibiotics, that's what happens, that the gut bacteria have gone, and then the empty places that are created, the pathogenic bacteria come.
Right.
So that's how the antibiotic induced diseases come that way.
Oh, okay. That's interesting. Like, It's interesting that the same location will be occupied by two different kinds
Yeah.
of bacteria, and we want only good ones to stay there and not the bad ones.
See, that's what you do in a disease like Clostridium difficile diarrhea. The patient is suffering from diarrhea and the antibodies are not working. So you give healthy fecal transplantation so that you bombard your gut with these healthy microbiomes so that Clostridium difficile is washed out.
Okay. Yeah. So then, your point, which you said, we should not take antibiotics just
Right.
randomly, is because
Yeah.
the same position it will be attacked, but then now instead of the bad bacteria, good healthy microbes are going to be eliminated from the gut. Right. We also have some questions, which are like, how does the microbiome change during menstruation time? And are there any studies which talk about, um, do you want specific microbiome during that period, or like before or after?
This is something which I'm not aware of. I'll have to check microbiome changes during
Sure.
menstruation, and this is something which I'm really not aware of. I need to check on that.
All right, sure. And are there any studies which talk about mental disorders? Like you mentioned that it's been shown that Alzheimer's or Parkinson's could be related. So like exactly what kind of relation? Because you also mentioned something about gut-brain axis.
Okay, yeah. So today these studies, see the gut microbiome. I mean the diabetes obesity started first, so we know a lot about molecular mechanisms involved in diabetes obesity, but mental disorders have entered recently. So today
Okay.
there are only associations. So you know what kind of microbiome alteration happens in Parkinson or Alzheimer's disease or even autism spectrum disorders, and people have shown the beneficial effect of probiotics on autism disorders. So but the molecular mechanism of how this happens is not known. There are some hypotheses, but it is exactly not shown.
But like in all, we know that since there is a gut-brain axis, then depending on what
Yeah.
your gut microbiome.
So, I mean, for example, how the microbes promote the formation of amyloid plaques and other things, there are hypotheses on that, but exact details so far we have not understood.
Okay. And then like to treat diseases, you mentioned some things about like how microbe-mediated drugs have been developed for various therapeutics and
Right.
diagnostics. So what exactly is done in that? And then can we have what kind of diseases are like being targeted, or we could
See, if you see that list, the diseases were mostly Clostridium difficile diarrhea, IBD, colitis, and some form of cancers and obesity. So these are the diseases for which such drugs are developed. For many diseases, the drugs are in the developmental stages. So, but so far, neurological disorders, I don't know whether any drugs are in trial at any level of trial for neurological disorders, because as I said that these are late entrants. Neurological disorders are late entrants and the interest has developed in last few years. Whereas diabetes obesity is there for ever since the microbiome studies started, immediate entrants were obesity and diabetes.
Right. Yeah. And then these are some things which are still in development, but
Right.
till all the medicines or drugs are developed, how can our normal lifestyle changes can be helped to have good gut microbiome? Are there any studies?
Yeah, I mean, as I said, two things. One is the good diet, which is high fiber, low fat, low sugar, and use of natural probiotics, natural fermented foods that are available. So even the yogurt and curd, which are naturally fermented, those indeed have beneficial effects.
Yeah. And then what about like different lifestyle changes, like doing exercise, getting up early in the morning, or How are they?
There are some studies on circadian rhythm and microbes, but those are very few. So the people also talk about effect of your life. I mean the daily cycle on the microbiome, but circadian rhythm and microbiome, there are very few studies. So how your daily life affects the microbiome in terms of your circadian rhythm is something which is still in infancy.
Okay, thank you. Um, we also have some question when you mentioned that your gut microbiome also interacts with the skin microbiome. So there was question like how does it affect and
See, I mean, it is again, these are all new observations, but the
right,
simple logic is that uh, the gut microbiome affects your metabolism, and in turn, the metabolism then is reflected in your skin secretions. So the composition of your skin secretion will be affected by the metabolism of gut microbes, and which in turn would change the skin microbiome. So those studies have done in, especially in diabetes, that how the alteration in diabetes changes the sweat composition and the in turn the skin microbiome.
okay, interesting. And like as a community, can we do something to promote or like help in the microbial research? Are there any, like you mentioned, there are 4 or 5 organizations who have started studying different animal, plant, microbes,
Yeah,
human
these are
microbes.
commercial companies. So these
Okay,
are commercial entities which are using microbiome therapies for business purpose.
right. So then, apart from the business part of it, is there anything on the community level that we could do to promote this
Yeah,
research?
so community level, the I mean, as the best thing at that should be done at the community level is to promote the healthy diet
Right,
and work on that, the promoting the healthy diet and healthy dietary habits. I mean,
right,
obviously, there are certain things which go without saying that when you talk about oral microbiome, the tobacco chewing or smoking badly affects the oral microbiome or upper respiratory tract microbiome. So all these things are interrelated.
right. I believe that I have covered most of the questions. Um, and I'm sure with more studies we'll get more
More,
data
yeah,
about
yeah,
this.
right, yeah.
One more question was how chemotherapy or different uh, therapies that have been given so far have been connected to how gut microbiome gets affected.
Yeah, so again, this is an upcoming field, and
Right,
the it is the some of the therapeutic agents do affect the gut microbiome composition, and one of the well-known example is metformin, which is used for diabetes treatment, that affects the gut microbiome. So there are plenty of studies on that. And another line of study which was being done abroad, not much in India, is the some of the chemotherapeutic agents, uh, they do alter the gut micro. I mean, they do alter the gut microbiome and resulting in discomfort in the gut and diarrhea kind of things. So some of the studies were being done to use kind of probiotics on such patients to improve their quality of life. So those
interesting.
studies are and also there are studies on changing the efficacy of the drugs using microbiome. So this was again study done on blood cancer, that in some cases the drug does not work on certain individuals, but if you
Hmm.
alter the gut microbiome, the way the gut, that drug is metabolized in the gut changes, and ineffective drug can be made effective. So those are the kind of studies those were done for some of the drugs.
Oh, that's
So a lot
interesting.
more is yet to come from such studies,
Right.
but definitely for chemotherapy, when there is the quality of the life is deteriorated because of these chemotherapeutic drugs, the probiotic kind of therapies help in improving the quality of life.
That's nice. And then, since all the individuals have different microbial composition and everybody has different capacity to digest or not
Right.
digest the same food, so how do we know, like, what is my microbiome? Are there any clinics or something which do diagnose that for you?
It's not yet started in India.
Okay.
But abroad, there are services. I mean, you register online and you get one courier comes. I mean, the package comes.
Mhm.
You put your fecal sample into that as per the instructions. Send it to this thing, and then they give you your complete microbial profile and also tell you about your nutritional status. It's a business then, because then they
Right.
recommend you diet or their own probiotics. So they say
Yeah.
that your microbiome is deficient in this vitamin, so you take this probiotic from us. So
Okay.
I think in US and especially in USA, there are many such companies.
Right.
We had worked with one such company for a while.
Okay.
They wanted to expand the business to India.
Okay, that's interesting. But till then, I think we can just stick with fiber-rich diet, low sugar, low
Right.
calories,
Yeah.
and
Yeah. Yeah.
do good exercise and see what we can do.
Right.
Thank you, sir, so much. If you
Thank
want,
you.
you
My
could
pleasure.
conclude like in 1 or 2 lines. We would like that.
So, I mean, my conclusion, my take-home message always for microbiome studies is that if you want to maintain a healthy microbiome, uh, have a healthy lifestyle, consumption of high fiber, low fat, low sugar food. Don't abuse drugs and alcohol, and especially for the I mean, not just for women, men also. For the younger generation, the advice is that unless it is medically called for, do not get carried away by the temptation to have cesarean delivery and not to breastfeed, because you are damaging your child's life by giving a cesarean birth and not having breastfeeding. So that is always been my message to everybody whenever I talk about gut microbiome.
Thank you, sir, so much for such a wonderful session.
Thank you. It was my pleasure. Thank you.
Saili?
Sorry, just a second.
Mhm.
So thank you so much, sir. We want to start with the acknowledgement section of the talk to conclude the talk. Thank you so much, sir, for such an informative session and for taking all the questions from our audience. We would also like to thank Jnanaprabodhini Foundation for being a huge platform for us to conduct such awareness sessions. We would like to thank iBRETA team for their support and their help to conduct and organize this session, and our support team, Dr. Swapnita, Revati, Mayur, Pritesh, Omkar, and Veena, for their constant support. A special thanks to all of you for joining us today and for your attention and your time. And I hope we all try to enhance our gut microbiome, and we have learned a lot through this session. So let's um, um, let's go home with the thought that our gut is our second brain. And thank you for everyone.
Thank you and wish you all the best.
Thank you, sir.